The cytoskeleton of eukaryotic cells is a dynamic network of protein filaments that provides structural support, facilitates cell movement, and enables intracellular transport. Two major components of this network are microtubules and microfilaments. Microtubules. Microtubules are rigid hollow rods with an approximate diameter of 25 nanometers. They are polymers formed from globular protein subunits called tubulin. Tubulin exists as a dimer composed of two slightly different polypeptide chains, alpha-tubulin and beta-tubulin. These alpha and beta-tubulin dimers polymerize end-to-end to form protofilaments. Typically, 13 protofilaments assemble laterally around a hollow core, forming the cylindrical microtubule. Microtubules have a distinct polarity, a plus end where polymerization is faster with exposed beta subunits and a minus end where polymerization is slower with exposed alpha subunits. This polarity is crucial for their biological function and for the directionality of motor proteins. Microtubules are primarily composed of alpha-tubulin and beta-tubulin proteins. They also interact with various microtubule-associated proteins, or MAPs, which regulate their assembly stability and function. Examples include kinesin and dynein, which are motor proteins and proteins involved in microtubule types based on function and location. Cytoplasmic microtubules are found throughout the cytoplasm. They are involved in maintaining cell shape, intracellular transport of organelles and vesicles, and cell motility. Mitotic spindle microtubules form the spindle apparatus during cell division, both mitosis and meiosis. These can be further categorized. Astral microtubules extend from the microtubule organizing centers such as centrosomes towards the cell membrane. They help to position the spindle. Polar microtubules overlap in the center of the cell, pushing the spindle poles apart. Kinetochore microtubules attach to the kinetochores of chromosomes, pulling sister chromatids apart. Ciliary and flagella microtubules form the core structure of cilia and flagella, known as the axoneme. They are responsible for cell movement or the movement of extracellular fluid. They typically exhibit a 9+2 arrangement with 9 doublet microtubules surrounding a central pair of singlet microtubules. Maintenance of cell shape and polarity, provide structural support and help determine the cell's overall shape and internal organization. Intracellular transport, act as tracks for the movement of organelles, vesicles and macromolecules within the cell. Mediated by motor proteins like kinesin moves towards the plus end and dynein moves towards the minus end. Cell division form the mitotic spindle, which is essential for the accurate segregation of chromosomes to daughter cells during mitosis and meiosis. Cell motility forms the structural basis of cilia and flagella, enabling the movement of single-celled organisms or the movement of fluids and particles in multicellular organisms. For example, think of mucus in the trachea or eggs in the fallopian tubes. Cell signaling plays a role in various signaling pathways by acting as scaffolds for signaling molecules.